Okay, here's where we left off. Now we know that cellular respiration is the term that describes a set of biochemical reactions that explain how living cells can very efficiently use oxygen and organic molecules like glucose in order to very efficiently create ATP, which is the energy for using our cells. And the example that we always use when we talk about cellular respiration is to use a molecule of glucose. It's very simple, and a lot of our cells really like using glucose, although they can use other organic molecules. So here is the basis. We start with oxygen and sugar, and through the magic of mitochondria and cellular respiration, we end up making these charged batteries, the energy for making life happen, called ATP, and we end up with waste products, carbon dioxide and water. Now, cellular respiration, we're going to use the example of a glucose molecule. And I think it's really useful to use these PowerPoints as your guide for what you really need to master to do well on next week's quiz. So first, we need to think about total of cellular respiration. There are three main components to cellular respiration. The first one is called glycolysis, and it takes place outside of the mitochondria, but inside of the cell. It takes place in the cytosol, right? So then we have got this gray thing. This gray thing is the mitochondrion. And the mitochondrion is where two of the three steps of cellular respiration happens. Step 2 is the citric acid cycle, also called the Krebs cycle. And step 3 is the electron transport chain. First, we're going to talk about aerobic cellular respiration. Aerobic means with oxygen, okay? Then we'll talk about anaerobic cellular respiration. Anaerobic means without oxygen. Okay, let's get going. Yeah, the citric acid cycle is also called the Krebs cycle, same thing. First thing you need to know for next week's quiz is the first thing you needed to know is what is the formula for cellular respiration, right? But that was last video. Now, next thing you need to know is the order of the three stages. What is stage 1? Stage 1 is glycolysis. What is stage 2? Stage 2 is the citric acid cycle. Or the Krebs cycle. Stage 3 is the electron transport chain. Next thing you need to know, glycolysis happens outside of the mitochondrion and does not need oxygen. Stage 2, the citric acid cycle, does not need oxygen, but happens inside of the mitochondrion. And stage 3 is the electron transport chain. It happens in the mitochondrion, and it definitely needs oxygen. Good. Now, let's move on to some more detail. Let's start by talking about the first stage, which is glycolysis. Glucose is a 6-carbon molecule. Its formula is C6H12O6. And it is going to be broken in glycolysis to make 2 pyruvic acids. So 2 smaller molecules. Each one is half the size of a glucose. By doing this, pyruvic acid is the same as pyruvate. By doing this, it makes a net of 2 ATP. This step doesn't need oxygen, and it doesn't need the mitochondrion, right? It also makes a little bit of NADH. Your red blood cells inside of your body, they don't even have mitochondria. They don't use up oxygen because it's their job to carry oxygen. So it's good that they can't use it. Your red blood cells still need ATP to stay alive. They use this very, very wasteful, inefficient way of making ATP. They use up a ton of glucose for the amount of ATP that they get. This is glycolysis in more detail. Glycolysis will net 2 ATPs. So it uses to make this unstable molecule, and that ends up making four. Anyway, just remember that it makes two, okay? And it splits one glucose into two pyruvates. Pyruvates are pyruvic acids. It's interesting that a little bit of water gets made in this step, just a little bit, not very much, but we're not using oxygen and we're not making CO2. Now, the second step is the citric acid cycle, which is also known as the Krebs cycle. The citric acid cycle doesn't make very much ATP. It does make a lot of NADH and some FADH. So the strength of the citric acid cycle is not that it makes ATP, but it does make molecules that are going to power the third step. So this is the second stage. The second stage has got two substages. The first substage you need to know, it's on the quiz. That pyruvate that was made by glycolysis, that will be broken down to create acetyl-CoA on the quiz. The first step of the Krebs cycle is this preparatory phase, and it's going to take the pyruvates made by glycolysis and turn them into acetyl-CoA. Now, this does release some CO2. It doesn't make any water. All right? Acetyl-CoA. So this is the second step of the Krebs cycle or citric acid cycle. This is the big thing, okay? And it's a really, really intricate series of biochemical reactions. This image you've got here, very simplified. So the acetyl-CoA gets fed in, and as the acetyl-CoA goes through all of these different steps, you'll see that a little bit of ATP gets made, some carbon dioxide gets made. NADH and FADH gets made. No water gets produced, some CO2 gets released, but all of this stuff is a really complicated series of reactions. Let me show you. Okay. I had to memorize this at one point in my life. I can't even imagine. Look, there's an enzyme that turns that into that. There's an enzyme that turns that into that. There's an enzyme that turns that into that. Yikers! You don't need to know it, okay? You need to know that the Krebs cycle has got two steps. Part A was taking pyruvate, turning it into acetyl-CoA. Part B is going to take the acetyl-CoA, run it through the Krebs cycle. Technically, two terms of the Krebs cycle are needed to dismantle our glucose, and it is going to generate some carbon dioxide, no water, but it's going to make FAD and NADH that's going to power the electron transport chain. It does make a little bit of ATP, so it makes ATP, it makes CO2, it makes FADH2, and NADH, right? From the point of view of the electron transport chain, this is going to power the electron transport chain. All right, now we get to the electron transport chain. Here is where the excitement happens. Right? The excitement. Here's a close-up of the electron transport chain. I'd like you to notice that for my lab and my quizzes, I will not hold you responsible for the kind of molecular detail on the electron transport chain that your SynBio activity did. The SynBio activity was very useful. It taught you a lot about how these molecular reactions work, and I think it creates a very detailed mental picture that can fuel your understanding of my simplified Reader's Digest kind of version. So the electron transport chain. The electron transport chain uses electrons that have a lot of potential energy. And as they get handed from one of those intramembrane molecules to the other, they give off a little bit of their energy along the way. At the end of of their tour, giving away little bits of energy, they need to be thrown away. And they get thrown away by being added to oxygen. So oxygen is termed the electron acceptor of this particular reaction. Without oxygen, these electrons would pile up. And as they pile up, they end up damaging the mitochondria and stopping cellular respiration. So we need oxygen to get rid of these electrons and carry them away in the form of water. The NADH and the FAD is going to power proton pumps. Proton pumps are going to pump protons. Remember, a proton in this context is the same thing as a hydrogen ion, same thing as a hydrogen ion. So these, the NADH and the FADH, they pump protons into this space. Now the protons are super, super, super crowded, and they're like, wow, we don't like being crowded. Now we've got this molecular machine, which technically is an enzyme, but a molecular machine called ATP synthase. And ATP synthase says, oh, you're sad. You're all crowded inside of there. Well, I'll let you leave. Protons are not allowed to go across the phospholipid bilayer, but they're allowed to go through the ATP synthase. But as those hydrogen ions, those protons, are leaving the crowded area, it's like they're going through a turnstile. And as they turn the turnstile, they're spinning the machine that allows ATP synthase to turn ADP and phosphate back into ATP, right? So these machines were powered by NADH and FADH, and they are going to pump hydrogen ions into this space. The hydrogen this image is flipped from the last image I just showed you, all right? Sorry for the complication. But now all these hydrogen ions are like, oh, wow, we're so crowded. We hate being crowded. And here ATP synthase says, oh, I'll let you leave. And as they go, yay, we get to leave from where we're crowded, they go piling through that protein just by the power of facilitated diffusion. And that is going to allow that enzyme to make ATP synthase. Okay? So the NADH and the FADH that was made by the citric acid cycle power the proton pumps. The proton pumps pump protons into this space. Now the protons are too crowded. They want to leave. The ATP synthase says, you may leave through me. And as they leave through ATP synthase, as they leave through ATP synthase, they power the creation of ATP. So the the electron transport chain is where we make most of the ATP. This image here is from next week's lecture on photosynthesis. And on next week's lecture on photosynthesis, we'll see another electron transport chain that uses different proteins, but the same logic, forcing protons into a space, making them crowded, allowing them to leave to power the creation of ATP. Okay? So step 3, the electron transport chain, this is where all of the excitement happens. We are not going to make CO2 at this step. The CO2 got made in the citric acid cycle. The citric acid cycle, the 2 stages of the citric acid cycle. But here we're going to make almost all of the ATP, and we're going to make lots of water molecules. So when there's oxygen around, we make a lot of ATP, and almost all of that ATP is made by electron transport. Almost all the water is made by electron transport, and the CO2 got made here at the citric acid cycle. Great, we're going to pick up there in our next video.